America? What do you watch, soccer or something like that? You watch real football? Actually, I used to play soccer. It's a lot harder. But I always get the feeling you just pick it up.
Scare up. Yeah. That would be her training.
And I just kept thinking, my God, just pick up, first pick up the puck and throw it.
That means this is
Yeah.
That's great. That's hilarious.
Let's get started. So today we're going to get most of the way through the visual system, and we'll finish up the rest on Thursday. And then we'll start getting into the auditory system. I think if I got it right, the next quiz will be, I don't have the date in front of me, but it'll be Thursday of next week.
It is.
And I think that's just enough time. We have to get another quiz in to get you guys sort of jump started again, but at the same time, we have to get it in enough so it makes a difference for the exam, which is not too long after that. So it'll be Thursday of next week. And I'll try to remind you again on Tuesday of next week too, so you have a week's warning. We went over a lot of material in the retina the last time, and I just wanted to revisit this one slide just to remind you of the circuitry again, and then we'll get on to the we'll get out of the retina. But remember that if you're trying to segregate the fundamental functions of those circuits that make center, surround, antagonistic receptive fields, the best way to break it apart is to think about what certain roles always are, and then how they switch depending on whether or not we're talking about an on-center versus an off-center receptive field. And what I said is, if you actually were building a retina, first you would put photoreceptors on the back of the retina pointing into the choroid substance. And their job is always just to become hyperpolarized in response to light. That's all they ever do. They don't care who they're talking to. They can talk to multiple receptive fields, it doesn't matter. And they don't bless glutamate. Then if you want to build receptive fields, you do that with horizontal cells, which are the blue objects here. And horizontal cells determine a lot of things. They actually construct the receptive fields. It's not on-center versus off-center. They construct the receptive fields. And first, and I didn't mention this in the last lecture, I just added this to the slide, one of the things they do is they determine where on the retina the receptive field is going to be. So if we're a bunch of horizontal cells, we pick a part of the retina, we say we're going to use those photoreceptors as our inputs and outputs. And then we arrange our cells so that our inputs are in the surround and our outputs are on the center. And our job is to simply do the opposite to the photoreceptors in the center that light does. So since light always hyperpolarizes photoreceptors in the center, the job of horizontal cells when light is shining on the surround is to depolarize them. And that's it. And what effect that has on on-center versus off-center has nothing to do with the horizontal cells. That's just the mechanism of antagonism. They also determine the size of the receptive field. They're larger, as we'll see, in the periphery as they are in the center. They also determine the shape of the receptive field, and as we said, they're always annular in shape, circular in shape. And you can see why, because if you're going to have a contrast between the amount of inhibition and excitation, or vice versa, in a receptive field to get contrast enhancement, you want to do that at all possible orientations of a change in contrast. So it's a circular receptive field always. They provide that mechanism of antagonism between the center and surround. And then, yeah, as I already said, they always do the opposite of what light does. So it's really the horizontal cells that are governing all of those things. And it's kind of a nice idea. It's as if the receptive fields have been prefabricated and are done by the time the bipolar cells come along, and they can just pick these prefabbed receptive fields and choose them as their center. So if you want an on-center bipolar cell, what it'll do is it'll choose the center of this receptive field that's already been built here. And then what it'll do is respond to less glutamate by becoming excited. That's the definition of an on-center bipolar cell. And ask yourself always, if it becomes excited by light, then it must be inhibited by glutamate, because it's excited by less glutamate, therefore it must be inhibited by glutamate. And the opposite is true with an off-center. And that defines really what the nature of the receptive field is. And it's the postsynaptic surface of the bipolar cell that defines all that, how it responds to glutamate, if it's inhibitory, excitatory. And then we ended this by just reminding you that these are not mutually exclusive receptor fields the way I used to think they were, and that's where the real complexity and elegance comes in. That, again, the photoreceptors don't care who they're talking to, and they can have, as the center of one receptive field, can also be the target of horizontal cells or the source of input to horizontal cells to form other receptive fields. There can be a tremendous amount of overlap between these. And the complexity of the interactions, the analog interactions within that is tremendous. And that's where most of the processing power comes from. Yeah.
Can a horizontal cell be part of two different receptive fields?
Yeah.
I mean,
Yeah, it could overlap tremendously.
I thought it was just in terms of the bipolar cells, in terms of the receptive fields they get to use. But horizontal cells can be double, like in some of those overlapping areas, you
Sure.
can have the same horizontal cell be part of the receptive field?
Mhm.
Okay.
Yeah, and that's where a lot of the elegance comes in, I think. And it could even be completely overlapping if you think about it. This receptive field could be constructed in advance. And one bipolar cell could turn it into an on-center, another one could come in and turn it into an off-center, and it's the same receptive field with the same horizontal cells, it doesn't matter. And that's where the elegance comes in, that's where the spatial resolution comes in. So I wanted to remind you of that, because I think that summarizes nicely what the construction of the retina is and what the different roles always are. It's always the case that these complex glutamate, it's always the case that the horizontal cells build the receptive field and uses the input, the surround, and the output as the center, and they just provide the mechanism of antagonism. The only thing that changes from center to, from on-center to off-center is the postsynaptic surface of the bipolar cells. And then as goes the bipolar cell, so goes the ganglion cell, so there's no special trick there. If you have an on-center bipolar cell and it hooks to a ganglion cell, that becomes an on-center ganglion cell, and there's no more complexity. Is everybody clear on that? Okay. So let's move back from the retina and start looking first at the sort of gross features and then the more detailed features of what happens, what type of processing happens as you move back from the retina into the lateral geniculate and the visual cortex. And the first thing that you would notice if you looked at the optic tract and you started stabbing every single axon inside the optic tract and mapping out its receptive field. And remember, the receptive field always by definition is whatever excites or inhibits the target cell that you're looking at. All these fibers are the outputs of ganglion cells. So if you stab a given output fiber and you map around, most of the areas of the visual field won't excite or inhibit it, and then there'll be a very limited area that will. And if you started mapping out those for each axon, what you'd find is that 20% of the retina is 20% of the axons, 20% of the retina that you map would represent 80% of the fibers within the optic nerve. And the reason is because 20% of the retina is about the foveal area of vision, where there's an enormous density and very little conversion. So 20%, I'm just picking these numbers up roughly, but 20% of the retinal surface would represent 80% of the fibers that you run into. And then 80% of the retinal surface would only be reflected in about 20% of the fibers that you ran into. And the reason there is there's much more convergence. There's many few fibers that are converging into, even though they cover a greater area of the retina, they converge into much fewer fibers within the optic nerve. You'd find out that all of the receptive fields that you mapped would have that annular shape, center surround antagonism. And then you start, if you did a more detailed analysis, you start noticing that there are slight differences, not slight, but rather large differences in the types of fibers that you ran into. The ones that are covering 20% of the retina are magnocellular fibers, and we just touched on that in the previous lecture. But they have the properties of magnocellular fibers as opposed to the 80% of the fibers or 20% of the retina that you run into in the optic nerve, which are parvocellular. And that gets its name, those names, by the way, those divisions, they sort of start, but they continue through the visual system. They start with the size of the ganglion cells themselves. The magnocellular fibers are fatter, they're faster, they have bigger cell bodies. They have bigger cell bodies because they're converging information. This is a picture of ganglion cells in the part of the retina that's in the periphery, which is where the magnocellular division dominates. And if you look at the size of the cell body, it has a much wider convergence, so it has much larger receptive fields. Therefore, they're much more sensitive to light, but they're much lower spatial resolution. And because they have fatter axons and fatter cell bodies, they're called magnocellular, as opposed to these other distinct fibers that are coming out of much smaller bodied ganglion cells that have much thinner axons or slower. And they're for the parvocellular division, and they're concentrated in the fovea. Foveal vision is dominated by, it's not exclusively shown here, but it is dominated by color vision, cones, as opposed to the magnocellular division, which has a mixture of cones and rods, but mainly rods. So it's a little bit of color vision, but mainly it's sensitive to illumination, not to color. There's a number of other distinctions between them. Most of which I've projected. Okay, well, I could get into what their projections are coming back. As you're looking at this, then you find that that's about it. That you have a magnocellular and parvocellular division is preserved religiously all the way back to the visual cortex. Now, as they project back, half of them have to cross over and the other half don't. And before we go back into what happens in the different areas of the visual cortex, let's look first at just the general projections. If you mapped out the receptive fields, the total aggregate of receptive fields of the magnocellular cells in the right retina, as has been attempted here, you find that what Netter's tried to do is map out quadrants in dark and then in a different color to show magnocellular versus parvocellular. If you were looking at just the right retina, you notice that it's offset a little bit, and that 80% of the retina is magnocellular, and the other is parvocellular. If you track that on back through the visual cortex, all of this would have crossed over to this side and projected itself along the, what's this fissure here? Cochran-Fisher. And it has, again, he's done a very good job at trying to map the upper and lower halves of the visual field as they project back through the lateral geniculate nucleus and then back to the Cochran-Fisher. The parvocellular division, foveal vision, projects back as well and tries to cross over to the other side. And it's actually trying to make life simple for you. If you think about it, the right retina, the nasal half of this retina is trying to project over to the left hemisphere. So the left halves of the retinas are trying to go to the left hemispheres. The right half of the retinas are trying to go to the right hemisphere. It makes total sense. The only thing that confuses students a lot of times is that if they're asked, where in the visual field does this project, it always has to cross over. But the idea, once you get to the retina, is that the halves of the retinas are trying to do the right thing. They're trying to make it simple. The left halves are trying to go to the left hemisphere, the right to the right hemisphere. The only thing that switches it over is all going through a lens. So if you're ever asked a question, which you probably will be on a quiz or an exam, if you're asked where in the visual field does something project in the cortex, always put it through the lens first and figure out where it projects on the retina, and then you'll figure out where it projects on the cortex. And that's true also with the upper and lower halves, that the upper halves of the visual field project to the lower parts of the visual cortex below the Cochran sulcus, but that's because it goes through the lens too. So if you figure out that it flips over so that the upper halves go to the lower halves and then the lower halves try to follow suit and go to the lower part of the visual cortex, it's simple. The only thing that seems complicated is it goes through the lens, so it gets flipped upside down and inside out. The rest is just straightforward when it comes to anatomy. And we'll go over this projection in a bit, but as these go back and do the logical thing, if the left halves of both retinas have to project to the left hemisphere, one has to go through the optic chiasma. So the nasal halves of the retinas are the ones that go through the optic chiasma so they can cross over and get to the correct hemisphere. The lateral halves are the ones that go straight back. the biggest difference between the magnocellular and parvocellular divisions as they project is that the parvocellular division is considered kind of an older part of the visual system. And I mentioned this, I think, briefly in the last lecture, that the magnocellular division, more primitive species or earlier species like dinosaurs, were considered to have only magnocellular divisions. They could only sense, remember, one of the properties of the magnocellular division is it doesn't sense color, but also it's very sensitive to motion. And for us, it's really, we're sensitive to motion because we want to detect things so we can bring our foveal vision, which is the parvocellular system, onto target to figure out what it is. With more primitive species that are lower down on the food chain or earlier in evolution, there was no luxury Cadillac system, there was no parvocellular system, there was just the magnocellular system. So they were mainly concerned about detecting motion, detecting objects that were moving in dim environments, so night vision. And there's other anatomical hints that unlike the Cadillac parvocellular system, the magnocellular system has to communicate with other lower areas of the brainstem and other structures. Unlike the parvocellular division, the magnocellular division, as it's coming back, as the fibers are projecting back, have a series of subcortical targets as well. And one of them is the hypothalamus. There's a right above the optic chiasma, a certain number of fibers project up into the hypothalamus, into what's called the suprachiasmatic nucleus, which is pretty easy to remember. And it's thought to regulate, and it still does in us, regulate circadian rhythms. It doesn't create binaural rhythms. We're doing some research on circadian versus binaural in my lab, so I finally figured out the difference between those terms. What binaural represents is something that repeats on a daily basis, but isn't necessarily synchronized to the light-dark cycles. A circadian rhythm, so that's created by clock genes and such. A circadian rhythm is one that can be reset and synchronized by dark light cycles. It's dependent on it. It's not created by it, but it's dependent on it. So when you go through jet lag, say, what's happening is when you're going through jet lag is the rhythms of your body, which are gauged by these binaural cycles, are being shifted. They're way out of sync compared to the light-dark cycle. And a lot of the confusion you feel and the maladaptation and exhaustion you feel is because they're out of sync with each other. That the clock genes, the binaural rhythm, is now out of sync with the circadian rhythm until it can get back into sync again. I find this fascinating that one of the thoughts is, and I think there's some proof to it, is that one of the earliest functions of the visual system, way back before it was even a high resolution sensory system, was mainly for that. That it was crucial for more primitive species to detect when the sun was out. And the reason is because they could detect and adjust their hormone levels in a binaural sense to prepare themselves to get up and do things versus go back to sleep. And that we still use it for that. So when our eyes are closed, they didn't have eyes. They had sensors that were near the skin, and the skin was thin enough to let the light through. And the original role of eyes was really just to detect the light-dark cycles. And we still do the same thing when we're asleep. We can detect when the sun's starting to come up, and well before we wake up, a lot of hormones start kicking in based on the fact that we're detecting light coming in through the eyelids onto the retina. And one of the solutions to the problem, I don't know how many of you, I bet none of you get up as early as I do. I get up at 6 o'clock every morning. Does anybody do this? All right. Get that coffee. Do you drink coffee?
I don't drink coffee.
Oh boy, that's really rough. So you get up at 6, you don't even
I
drink
get
any
up at
coffee.
5.
Oh my God. I used to get up at 5 and I would go down and exercise on exercise. I was a Marine, and then I've just gotten lazier and lazier with time. I can't even get up that early anymore. But I find that more and more when I wake up, my alarm goes off and I hate the thing because it always wakes me up in the middle of REM sleep. In a dream, I feel spaced out. It takes a pot of coffee to get me going in the morning. That's why you're probably doing so well without the coffee, because you're used to getting up early. But I bought a gadget which really worked, and the only reason I don't use it is because my wife hated it. It's a light that starts coming on. You give it a range of times you want to wake up, and then what it does is it starts coming on very, very slowly. It increments the light up like the sun's coming up. And in the wintertime, it was working miracles with me because it would slowly come on.
and I feel that the sun was coming up, it's summertime, and so I'm sure I wake up. It had an emergency thing. If it got to the end of the cycle, then it would just go off and wake up anyway. But it was wonderful. I mean, it was only a difference of like 15, 20 minutes and when I would actually get up. But it was all the difference in the world because it was presenting light that was making me physiologically adjust to the fact that the sun was coming up and I was getting ready to rise. So that system really does exist. And it really does affect you. There's other ways to do it. There was a guy, there was, I think he was a British neurophysiologist, and he wanted to find out the difference between binaural cycles and circadian rhythms. And so what he did is he went down into a cave. He conducted an experiment where he went down into a cave for like a month, and he had his colleagues adjust when he was fed and such, so he had no idea when the sun was coming up and going down, and there were no external clues coming into the experiment. While he was down in the cave, his wife divorced him. None of his colleagues, thank God, told him while he was down in the cave. That would have been an interruption in the experiment, but he was divorced. That's how dedicated he was. And through all of this, what he discovered is that when you're down in a cave and you have no adjustment to light cycles, you still have a binaural rhythm, but in his case, it went off by about 20 minutes. And that was it. And I guess he got tenure for it, but he lost his wife. He came out of the cave a changed man, I'm sure. But I guess he got a publication out of it. But so that's one of the projections on the way back. The magnocellular division deigns to talk to the suprachiasmatic nucleus because that's one of its earliest jobs was to adjust things and turn binaural rhythms into circadian rhythms. Another target as the magnocellular system heads back is it doesn't have just a privileged path to the visual cortex. It indeed goes through the lateral geniculate and gets information to the visual cortex. But it also drops down to an area called the pretectum. Remember when we were talking about the mesencephalon, we were talking about the pretectum. The pretectum has to do with pupillary reflexes. So it is the fundamental fiber path that gives you adjustments for different light levels to adjust your pupillary reflexes. And then finally, it has a large projection down to the superior colliculus. And remember we were talking about the superior colliculus. It used to be thought to be a purely visual tracking system. And then it was found out that it also receives auditory information. We'll find out later some sensory information for tracking objects in multimodality realms. But its primary function is visual tracking. And that's another subcortical target that's magnocellular, much more than parvocellular, and the reason being, again, it suggests what the magnocellular division is all about. It's for detecting objects and getting the eyes on targets and tracking where they are in space, but not really figuring out very carefully what they are. And then the projection back to the visual cortex. The parvocellular division, which is more the Cadillac, the newer system, its job is much lower, it's much higher resolution, but it's much slower. It has color vision as opposed to just high sensitivity detection. It has much smaller receptive fields. Its job is to figure out what things are. So we see something in our peripheral vision, which is magnocellular, we immediately bring the parvocellular onto it and foveate the object to figure out what it is. And that system has privileged access right back to the visual cortex through the lateral geniculate nucleus. And it doesn't touch go. It doesn't go down to any of the subcortical structures. Its job is to send information right back to the visual cortex to process it. We'll get into these other structures, but before we leave this general projection scheme, which I kind of like, so this is the visual field of all the axons if you added them together. This would be the total visual field of the left retina. This is the visual field of the right retina. These areas where they overlap are binocular, and these areas on the sides are monocular. Why do you think those areas exist? It's a really stupid answer.
Evolution is lazy.
What?
Evolution is lazy.
Well, that's not a stupid answer. That's an elegant answer, but it's not the right one.
Okay. I'm just assuming maybe.
Dynamic range.
Something like that.
No. Why do you think there's a clip of your visual field on either lateral extreme? Yeah.
Noses in the way.
Exactly. That's the only reason, there's no exciting functional reason for it. Is your nose is in the way, you can't see through your nose. So there's a cutoff here on the sides where this is the part where you can't see through your nose. So this is trying to see over here and you can't, and that's creating these parts of the receptive field that are just monocular. Yeah.
This is kind of random, but is it true that babies can't see in color? Like early in their development? Because I heard that, but I feel like true, like they should have cones, right? Already.
I don't think that's true. I'm trying to think, I vaguely remember some experiment about color vision in babies, and at least at a very early age, they have color vision. There may be a stage at which the Roslin cones haven't developed clearly, but I don't remember. The what? Yeah, yeah. I don't think there's a stage at which, particularly in the retina, I don't think there's a stage at which at least the magnocellular products are distinct from each other. Maybe babies don't appreciate the color vision as well, or the color contrast hasn't been improved back in the visual cortex. There's a lot of work that's going on, both in terms of lateral geniculate and the visual cortex, which is processing color vision. But I can't think of any experiments where they've gone young enough, at least, to find out that babies don't have color vision. Other questions so far? Okay. Anyway, I like the way that Netter's laid out these colors and these quadrants because it makes it easy to track things back. Obviously the retina is flipped over, it goes through a lens. As you get back to the visual cortex, and we'll say more about that later, if you look at the projection of these different areas, remember there's a retinotopic projection that's upside down compared to the visual world, but right side up compared to the retinas. But if you look at the posterior to anterior projection of the retina onto the visual cortex, you can see that the brown and black areas, which are foveal vision, are taking up about 80% of the visual cortex. And the other areas, which are the magnocellular division, peripheral vision, are taking up only about, in reality, 20%. Why is that?
The number of fibers?
Yeah. There's a number of fibers. You know, if you think about it, 80% of the fibers are coming back from the fovea. Then 80%, there's no optical constraints on the visual cortex. In the retina, if you want to have higher resolution vision, you have to pack a lot more density of receptors with smaller receptors than the fovea. So there's an optical constraint. And therefore, 80% of the fibers come out of that very small area of the retina. Once you get back to the visual cortex, there's no optical constraint, so you just expand out as much as you have to in order to process the information. So that the foveal vision is not only laid out in a much larger fashion on the back of the visual cortex, but a majority of the visual cortex, primary visual cortex, is devoted to foveal vision, and then it projects progressively from the center foveal to the peripheral vision as you go more and more anterior. That's the layout there, and then it's upside down, as I already mentioned. That's called, by the way, it's unfortunate to introduce this term in a visual system because it makes it a little bit confusing. That's called cortical magnification. It has nothing to do with optical magnification. We'll see it in the somatosensory system. We see it not really very much in the auditory system. But cortical magnification simply means that a small area of a receptive surface is magnified in its representation in the cortex for the very reason in the visual system, because it has to be magnified because it has to process twice, 80% of the information. So it just devotes more real estate to that compared to the other part. But that's cortical magnification. We'll see in the somatosensory system that areas of high density, say on the fingertips, of high density receptors will be cortically magnified in the cortex to look enormously large if you drew a homunculus showing the representation of the hand. And that's not optical magnification, it's because a small area of the receptive surface is being expanded in the cortex to cover a large area of the actual cells that are devoted to processing it. What else do I want to say about this? Yeah, that's about it. So let's go back and look at the processing that happens in the lateral geniculate, and then go back into the visual cortex where things get a little bit more complex, and continue to divide the magnocellular versus the parvocellular divisions as we go back. But if you look at the lateral geniculate nucleus, this is a nice cut through the thalamus. Here's the lateral geniculate that's been cut nicely.
Here's a nice stained section of the Lowers nucleus. The first thing you notice about the LGN is that it has a bunch of layers that have nothing to do with the layers in the cortex. The cortex is a laminated structure, also it has 6 layers, but this has nothing to do with the 6 layers of the cortex. If you look at these layers, the outer ones, let's call these, let's start here, 1, 2, 3, 4, 5, 6. The outer 4 are devoted to the barbocellular division, so it's not surprising that there's so much more devoted to it because it's 80% of the fiber. The most medial, if you will, or ventral layers, these two, are all the magnocellular division, and even then you can see that the cell bodies are fatted. So that maintenance of fat cell bodies in the magnocellular division continues on back into the Lowers nucleus. And if you look at this in a more schematic form, you can see that the first 2 layers are magnocellular, the outer layers are parvocellular, but also they alternate. The separate layers alternate in terms of the ipsilateral versus contralateral eye. So they're separated from each other, and they seem to switch over the order. So in the magnocellular division, the first layer is contralateral and ipsilateral, then ipsilateral, contralateral, ipsilateral, contralateral. What's weird about this is if you mapped out, this is a cut, it's like cutting through an onion. So you've done a coronal section, if you will, of an onion. If you actually mapped out the representation of the retina, roughly, on the other dimension into the slide, if you started poking cells in the outer layer, you'd find out that they're topically organized. They're not just randomly organized, they have a representation of the retina, at least that part of the retina. And then if you mapped out this layer, you find out it has a representation of the retina also. And if you can see through the outer layer to the next layer, you see that they're in register with each other. And then if you went deeper, this is approximate obviously because they're not completely the same area as the retina, but if you went to this, you find out that as you poked an electrode in the outer part of the Lowers nucleus, and say you found the cell, and you figured out where in the receptive field it was, and then you poke deeper, you find another cell, it would be in register with that one. And then if you poke deeper, let's pretend it's precise, you find another cell that's in exactly the same area. It's as if the receptive fields have been lined up with each other very, very nicely. Yeah?
See with the parvocellular layers, you've got redundant layers of the LGN. Is that the case?
Right.
So the outermost layer and the third layer are the same receptive fields, the same eye.
They're overlapping to a great extent. I'm lying a little bit. They're not perfectly in register, but they overlap more than they don't overlap. But yes, I mean, I don't know if I'm answering your question, but this is from the same part as this, but it doesn't completely overlap. All right, so they're massively overlapping, but they're segregated to some extent, but they're less segregated than they are overlapping and in register.
So I can understand why you'd have overlap between one eye and the contralateral eye.
Yeah, me too.
Same receptive field. Why have 2 representations of the same eye?
That's a good question. The other good question is that they don't talk to each other at all, which is very interesting. If you think about it, why wouldn't these layers speak to each other? It looks like an optimal place for binocular convergence, right? If you have a bunch of receptive fields that are laid out in register with each other approximately in the retinotopic organization, particularly between the 2 eyes, if you went all the way from this layer to this layer, the overlap's not as great. But the overlap between this layer and that layer, you go ipsilateral, contralateral, is tremendous. Why would you lay out a structure this way and then have no communication between the layers? Who would build it? I mean, question this stuff. This is stupid. I mean, anatomy is trying to tell you something. It's trying to say, look, I went to all this trouble to lay out these layers and register with each other. They must want to talk to each other. And if they wanted to talk to each other about the only thing I can imagine they'd be trying to do is binocular vision, because they're segregated so carefully. Not only in terms of magnocellular and parvocellular, but they're segregated very carefully in terms of which retina they're coming from. Why are they so separated in terms of layers, and then set out and register with each other, and then don't talk to each other and don't do any binocular vision? Same reason? Yeah. Yeah.
Well, don't they then go on to the cortex where they do converge?
vision, so maybe just it makes sense to division everything up neatly before you go on to the next integrating cells.
I would agree with that. I mean, if you got back to the visual cortex and you needed that segregation coming into the visual cortex, which is sort of true.
Because it does get organized.
Right. It's sort of true, but then why bother to lay them out so carefully in retinotopic organization? Those are mysteries to me. I mean, why have it so you can stab an electrode down through a series of those cells and have them all respond to the same area of the retina, and yet they don't even bother to combine information? I think it's one of the mysteries. We'll get at that a little bit. It's one of the mysteries of what the lateral geniculate does. I would submit that it has a lot to do with studying the lateral geniculate nucleus in animals that are under anesthesia. And there may be a lot more interaction than we know about, and that disappears. That's so often the case, it disappears under anesthesia. But I just can't accept that. I can't believe that they would be lined up that carefully and have absolutely nothing to do with binaural vision, binocular vision. Another mystery that's really surprised me about the lateral geniculate is that I used to think that the lateral geniculate nucleus, as usual, the insulting view of the thalamus is it's a relay nucleus back to the cortex. I used to think the lateral geniculate nucleus was, its only role was to relay information, visual information from the optic tract back to the visual cortex. It turns out that only 20% of the fibers in the lateral geniculate nucleus are coming from the optic nerve. Only 20%. Most of the fibers, remember we said there was a 10-to-1 ratio between corticothalamic fibers and thalamocortical fibers? Most of the fibers that are coming into the lateral nucleus are coming back down from the cortex. What are they doing?
Memories.
The what?
Memories.
Memory?
Yeah.
That's interesting. So how would they do that? They would be recycling or reverberating in a sense with the information that came in from the thalamus, or what's happening? I mean, what's going on with those fibers that are coming down? Why are there 10-to-1 fiber ratios? Yeah.
So the eyes are always saccaded, right?
As what?
The eyes are always moving. So
Right.
you have a fixed image of the
Uh-huh.
moment. Maybe they're doing comparisons from one moment to the next to see what has changed and where the update is.
It could be, yeah. It could be that there's a time lag behind what the signals left in the lateral geniculate versus the cortex, and the distinction temporally between them could resolve that to some extent. Yeah.
Didn't you say there's like visual input, that there's a sensory integration, so there's input from the cortex back to the thalamus?
It's certainly that, and it is certainly, not just in the visual system, but it's certainly true that the thalamocortical corticothalamic loop is crucial to processing in the cortex and the thalamus, always. It is known to be crucial. The problem is we don't know what in the hell it's doing. And I can take some stabs at it. Part of the reason, again, gets to anesthesia, I think. There have been studies that have been done where you can stab a cell in the lateral geniculate nucleus, and you can map out its receptive fields, its response properties, everything else you can possibly think of. And then you can ablate the area of the cortex that actually projects back down to that cell. And you can't detect a change in the response properties of the cell, which is kind of depressing if you think about it, if you're trying to figure out what happens, if there's any interactions at all. Why is it that you can actually ablate the area of the cortex that projects to it, and you can't detect any variation in the cell's response properties? It responds to the signal inputs that you were giving it before as if there was never a cortical input. I think what's happening, and other people do too, this is not just my theories, I think again, these are one of these simplistic views that are given, or enormous question marks that are given, they're almost like straw dogs because we're looking at animals that are under anesthesia. And that there's a tremendous impact and very complex impact between corticothalamic projections and thalamocortical projections that has a lot to do with processing and refining the image or whatever, and that we're not seeing that, and there's no change because the animals are anesthetized. And so much of the function is being suppressed by the anesthesia itself. I think we'll see other examples of this later on. My stand is that what's going on, and it's an important principle to understand, is that we don't passively receive information from the outside world. When we see something, it doesn't just come in through the lateral geniculate nucleus and then go to the cortex And then the cortex says, thank you, thalamus, I've got the information now, I'm gonna process it, get out of here. And maybe the cortical thymic fibers are just handshaking, saying, we got the information, now get out of here. We don't passively process information from the outside world. We test a series, let me see if I can say this clearly. We actively process the information. In other words, we're constantly manipulating the information that's coming in to test hypotheses about what it actually is. So if the signal comes into the visual cortex, and at a given millisecond in time, the cortex has a hypothesis about what that signal may be, what it can do then is project a patterned, very intentional piece of information back to the visual cortex to manipulate, and back to the lateral geniculate, to manipulate the nature of that information coming through. And if it goes one way, this hypothesis is proven. If it goes the other way, the other hypothesis is proven. In other words, we're hypothesis testing all the time about what's coming in. We're not just passively receiving it. And that interaction, those projections back to the thalamus, are in a sense manipulating the nature of input. You give me an input, and if you're the lateral geniculate, I get the input and I start asking you questions. And as soon as I start asking you questions, if I start asking you questions about the nature of the input you just gave me, in other words, if I start manipulating the nature of the input that you give me, I get a much clearer answer about what the input is. And if I'm not allowed to ask questions, I can't do that. Who's confused by that? I think that's the major function. It is that we don't passively receive it. We iteratively ask questions about what the nature of the input is. We interact with the outside world all the time. And one of the major places where we can interact with it is at the lateral geniculate nucleus in this case, where we get information to the visual cortex and come back and manipulate that information. And we'll return to that idea of the sensory systems as well, but that's the best I can do to think of what that function is. Many of the inputs are actually from the reticular activating system, vis-a-vis the interlaminar nuclei and the thalamus, I believe. What's going on there? What's a reticular activating system in the mesencephalon? It's arousal, right? It has to do with modulating input. Why is so much of the fibers coming into this area coming from the reticular activating system? It must be a perfect bottleneck to modulate the efficacy of transmission from the optic nerve back into the visual cortex. That's the crudest interpretation of what that may be, but it's a good one, I think. That this is a perfect place, if you want to adjust the efficacy of communication through the lateral geniculate nucleus, say to the expense of auditory information going to the medial geniculate nucleus, this is a perfect bottleneck to do that kind of adjustment, a perfect faucet, if you will, to adjust that efficacy of communication between the thalamus and the cortex. So if you get down to, and this will become a kind of repeated story for other thalamic nuclei, if you just look at it crudely, indeed the lateral geniculate nucleus is a relay nucleus. It does relay information up to the cortex. It does have center surround antagonistic receptive fields, now done neuronally with inhibitory interneurons. So it sharpens contrast to some extent. It can't be its major role, but it does that. I think this is the most interesting one. It is the first way station within which the cortex can manipulate its own input. And in a more fancy way, that's the first place where it can manipulate its input to ask questions about what's coming in and test hypotheses about what's coming in and what things are. And then finally, in a very crude interpretation of what selective attention could be, I think it's manipulating the efficacy of communication between the optic nerve and up to the cortex. So it's a good place to, if you certainly can attend more to what I'm saying than what you're seeing, and vice versa. So we know that we have this ability to sort of selectively attend to one modality versus another. Many, many areas of the cortex and other parts of the brain could be participating in that. But one of the areas could be in this nice bottleneck where you could actually sort of adjust the gain, if you will. I think it's much fancier than that, but that's about the best we can do now. So that's going back, that's about the best I can do with the lateral geniculate nucleus as a thalamic relay. I think that mystery of why it's tongue-in-cheek, I think we're going to find out a lot about what its function is and what these interactions between the layers are. One thing that will go beyond that question I raised in the last slide is that now we know, these didn't used to be recognized, but there are these layers that separate these two magnocellular layers and these four carbocellular layers. there are these layers in between which have been stained strategically, and you can start to see cell bodies in them, called the coniocellular layers. They weren't even recognized before. They were just, there were separations, identifiable separations between the layers. It turns out that the coniocellular layers do reach out in between the different adjacent layers of the lateral geniculum. Specifically, they target ipsilateral and contralateral layers, ipsilateral and contralateral layers, back and forth. They reach out. And if you can get in there and actually study the response properties of the coniocellular layers, they all seem to be very much binocular. So there is binocular processing happening in the lateral geniculum nucleus. And it's probably being done mainly through these coniocellular layers, which are reaching out. And that is part of the answer of what is going on about alternating layers and whether in retinotopic register is that these cells give the capacity for some interaction between the layers, which is interaction between the two retini. That's about as far as it can go, though, and as far as it's understood, again, because these are studied under anesthesia. Any questions? Okay. Let's head back towards the visual cortex. Now we're getting into 6 layers, and these are the standard 6 layers of the cortex. If you took a little chunk out of the, let's say, the primary visual cortex, and blew it up, you can see again, you remember these, there's the supragranular layers, 1 through 3. And then there are the granular layers, layer 4, which is extremely thick in the visual cortex, because there's an enormous amount of input coming in from the thalamus. And the granular layer is the input layer of the cortex. So in sensory cortex, particularly visual cortex in us, we have this really thick granular layer because it's got a tremendous amount of input coming in from the thalamus. And then the infragranular layers, as usual, the output layers of the cortex. This is oftentimes called striate cortex because it was some of the earliest cortex to be recognized as being so striated in terms of its 6 layers because it's got that thick layer 4. Balliard, I don't know what his first name was. Balliard discovered this really thick granular layer, so this is oftentimes referred to as the band of Balliard, which receives dense input from the lateral geniculate nucleus. What if you had absolutely no idea? You've had some beginning discussions about what the visual cortex does, but just pull yourself back a little bit and imagine what it would be like if you had to start doing crude experiments to figure out how the visual cortex is organized, and if you figured out how it's organized, you might get some sense of how it functions. And you did. These were early experiments that were done. What if you took a monkey and turned him into a little camera, and you put a hood over his head, and then you injected 2-deoxyglucose, which is one of the imaging techniques we talked about at the beginning of the semester. So you have a radioactive glucose. If you have cells that are excited and absorb the glucose, they become radioactive, and then you can whack the animal and then slice the areas of the cortex that you think may be activated, put it on the slide, and look at how it's been activated or unactivated. Some of the earliest experiments that were done on the visual cortex, in this case in the monkey cortex, were like that. And in this case, what they did is they sewed one eye shut and left the other eye open. And then they exposed the animal to light, the monkey to light. And then they whacked the animal and looked at the cortex. This is a coronal section through a piece of cortex. This is actually a band of white matter in the middle, and the gray matter is actually wrapping around that part. But if you look at the organization, white here shows the areas that were activated by just one eye. And you might get an idea. That's interesting. It looks like there's almost a column. It's not quite as obvious here, but it looks like there's almost a column right in layer 4 of lit up regions that were activated, interdigitated with areas that weren't. And the column actually would extend above and below, but it's really here in the middle where the densest activation was. You have these alternating columns of tissue that are activated by one eye. And you would have come up with 2 things at once. One is that there may be a columnar organization to function, that the columns are organized and they're interdigitated. So you might call this an oculodominant column, which it was. If you could actually, the same animal in this slide, if you could close that eye and then open up the other eye and expose them again, all these columns would exactly move over. So the areas that are dark here would become light, and these would become dark. And then you get a sense, my goodness, these ocular dominance columns are alternating back and forth between one eye and the other. Yet there's a columnar organization of the cells, I don't know what they're doing, but ocular dominance, one retina versus the other, is clearly still being segregated within the visual cortex, as was suggested before. Even though there's interaction at the lateral geniculate nucleus between the two different retinas, there's a strong segregation of the two different eyes. And that would be one dimension that you might derive from just these very simple experiments. Then if you did the same kind of experiment, but you changed the type of stimulus to different orientations, so you left both eyes open. And then you started looking at what happens if you just expose the monkey to vertical bars versus horizontal bars versus any bar orientation in between. You'd realize, I don't know why you would have done this experiment. You'd do it by accident, and then you come back and say you did it on purpose. But if you expose the animal to, say, vertical bars, you'd find out that the columns on another dimension aren't dominated by one eye versus the other. They seem to be dominated by the orientation of lines of the stimulus. And if you could do this over and over again for different orientations of lines, you find that these would shift over very slightly each time. So there's an ordered progression, still a columnar organization, but organization of function that doesn't have to do with ocular dominance. It has to do with the orientation of lines, and you might call these orientation-specific columns. And they would be in order, gradually moving over as you change the orientation. They're not randomly organized. So vertical would start here, moving all the way over to horizontal and everything in between. Having done those experiments, you would have had basically what was the earliest understanding about what the organization of the visual cortex is that it's segregated still in terms of ocular dominance columns that extend from the surface of the cortex down to the depth, to the white matter. And within each one of those ocular dominance columns, you get a sense of subdivisions of orientation-specific columns. None of this was actually discovered this way. It was discovered by, anybody know who? I'm going to call them Hubel and Weasel, and I'll call him Weasel for a good reason, as you'll see. It was discovered by Hubel and Weasel, and they were doing experiments at Johns Hopkins at the time. Other people had gotten Nobel Prizes for discovering they're discovering different layers of what the retina did and what the receptive fields of the retina were. Then there were Nobel Prizes sent out for people discovering what the receptive fields of lateral geniculate nucleus were. What Hubel and Weasel were doing is going back to the visual cortex with their microelectrodes and attempting to replicate the same thing. They wanted to find out what happens, what is the organization of the visual cortex as opposed to the lateral geniculate as opposed to the retina. And they would stick their microelectrodes from the surface down to the depth. This was not possible. They were very small electrodes. And they would have these experiments. I used to have a little movie of it, but it took too much time to show. But it was hilarious, where in these early experiments, they would take a paralyzed cat, and they would stick an electrode in the visual cortex, and then they would try to put little dots on the screen. So they had this ancient slide projector where they could make a slide that was black and had a little hole in it. And they could move this slide around, and it would move the dot around on the screen where the rat was fixated, and the cat was fixated looking at. And they were trying to map out the receptive fields just like the lateral geniculate and just like the retina, so they're using a dot. And they kept searching around, and they couldn't find any area of the visual cortex that responded to this stimulus. And so they would go back to the lateral geniculate, and sure enough, it responds to the stimulus, and they go back to the visual cortex. They couldn't find any areas of the dot that would make any difference in the cell firing in the visual cortex. Then by accident, they were pulling the slide off. They recreated this for the movie. It was funny. They were pulling the slide off the projector while they had their electrode in one of the cells of the visual cortex. And as the edge of the slide went off, the cell just exploded with activity. It was like, holy shit. And then they went back, and sure enough, every time they would take the slide and move it back and forth across that one area of the visual field, create a line, they discovered that was what the visual cortex seemed to like. It wanted an edge, it didn't want a dot. That's called serendipity. Has anybody heard of serendipity before? How many people have heard of the scientific method? And how many think that scientists really follow the scientific method? come on, you're young enough, you should think this. Nobody follows the scientific method. Nobody. If they do, they're the most boring scientists in the world. What happens is, you think I'm kidding? I mean, most of the most interesting discoveries I ever made were made by mistake. Serendipity, I'm not going to go into the history of the term, but serendipity was, if I remember correctly, there was some island in Greece and there were a bunch of naked mermaids on it or something. And sailors would run into this island by mistake, and then they go off and they try to explain to other sailors where it is because they wanted to find out. And you could never define where the island was. You had to discover it by accident. And it was the island of Serendipitous or whatever, I can't remember the name. The myth is actually much more interesting than that. But the idea is that when you discover things by mistake, those oftentimes are the best discoveries you ever make. And part of the reason is because you never intended to make them in the first place. So you always go in. The idea of the scientific method is this, right? You're supposed to form a hypothesis, right? Is that true still? You form a hypothesis. Then you form a null hypothesis. And essentially what the null hypothesis means is I'm an idiot, I'm totally wrong, and that's what I want to prove in my life, right? And if I can't disprove the null hypothesis, I'm not going to get tenure. I'm going to lose all my grants. My lab will be bankrupt. But I'm an honest human being and a scientist. I'm going to try to form a null hypothesis, and only if I can disprove it will I be correct. Right? No scientist ever does that. Am I correct? You guys are graduate students who are getting into science. No one ever does that. No. If you form a hypothesis, the reason for forming a hypothesis is to get you off your ass from sitting in your chair and actually go into the laboratory and find out what's actually happening. And the best scientists are ones, that doesn't mean you're going to succeed, but you form a hypothesis in order to go into the lab and start, well, they call it experimenting, I call it fooling around. You start fooling around in an educated way to see what's going on. And the best students, actually students oftentimes suffer from blinders and that they suffer from the hypothesis. Good scientists discover great things regardless of what hypothesis they happen to suffer from at the time that they're doing it. But graduate students oftentimes will go in and they're suffering heavily from this hypothesis. And when it doesn't work out, which it never does, you always try to design the hypothesis to be as simple as possible so you'll score. And it never works out because nature is really sassy and nature will just smack you around every time and tell you what a fool you are. Nothing will come out. The difference between an experienced scientist and a beginning scientist is a beginning scientist considered that a failure. And they get all depressed, my hypothesis is not being proven, I'm never going to get my degree, et cetera, et cetera. When you get some experience, you start realizing this always happens. And what you should do is start looking around in your peripheral vision in a metaphorical sense. That when things aren't working out, look around to what is working out. And that's where the fun is. So use your peripheral vision. When nature's saying, idiot, then nature's also saying over here, but look at what's happening over here. The thing you think is a failure is actually what's going on. And if you learn to turn your vision towards that, and foveate what is happening on, that's where the great discoveries are. I think some of the best papers I've ever had were ones where, in honesty, we were just fooling around. I could tell you 100 stories, but I won't. And then we stumble on something, and then we focus on the thing we stumbled on, and that turned out to be a really important discovery. And then, of course, you write the paper like it's what you were looking for all along. I mean, you're not going to say, I was just picking around, I stumbled on this thing, and isn't that cool? You always pretend like that's what your hypothesis was, and that's what continues to perpetuate the idea that the scientific method is actually used by scientists. It never is. Does anybody here repair their cars? When you're doing car repair, if you hear a banging sound in your car, in your engine, you'll pull over, right? And you'll open up the hood. But do you form a hypothesis and then a null hypothesis? I think, there's a sound and banging around my car. Maybe there's a nut loose. And I guess that's a hypothesis. But then I don't form a null hypothesis, maybe there's not a nut loose. and maybe I'll conduct a series of controlled experiments to find out. No, I form a hypothesis, I think there's a nut loose in the fan housing, I start fooling around. I usually break my engine, and then some nut drops out, but it was in the wrong place. And then I go back and pretend that's what I was looking for all along. That's really the way science goes, it's like car repair in that sense. And having said that, that's probably the most important thing you'll learn in this class, by the way, besides drinking coffee. What these guys were doing is fooling around. And they did have a hypothesis they were suffering from, and that's that they would be able to replicate and steal the work of other people who got Nobel Prizes back in the Louder-Nicollet nucleus and the visual cortex by using little dots, and they map out the receptive field, and everybody would be happy. And then by accident, by serendipity, they pulled the slide off, a line went across, and suddenly there was an aha phenomena that they would have never intentionally done in the first place, which is that what the visual cortex likes is lines, and it really responds to lines. It doesn't respond very well to dots unless you organize them right. And then they started trying to map this out, and they would stick their electrode down in a certain place and try to, as best possible, go vertically down from the surface of the cortex down to the white matter. And they'd isolate cells in a given area. So this is just a given area of primary visual cortex. They'd stick a cell and they'd map all over the place with their slide at this point. And to find out, with little lines at this point instead of just the edge of a slide. And they find out an area that the cell really responded to. And then they turn the orientation of the line back and forth until they got the maximum orientation of the line. So they got this notion that these are not just responding to lines, but they like lines of very specific orientations. And then as they jammed their electrode down through the different layers of supergranular and then granular and infragranular layers, they find out that all the cells that responded to a line, say, in this case of 45 degrees, were like lines of 45 degrees. And they came up with this notion that there's a columnar organization to the visual cortex where each column is interested in a line of a certain orientation, and it's interested in a line of a certain orientation all the way down to the white matter. So it's a similar function all the way down from the surface to the depth. We'll get back to this in a second. They got a Nobel Prize primarily for that discovery, that the organization of the cortex, not just the visual cortex, but of the somatosensory cortex, and we'll see in the auditory cortex, one of the fundamental organizational principles of the cortex is that it has a columnar organization from the surface of the cortex down to the depth, that each column is like a processing module, and they're adjacent to each other and they talk to each other. But that columnar organization of the cortex was one of the things that drove their Nobel Prize. The reason I call them Husel and Wiesel is that Wiesel is his name, Wiesel was working in Johns Hopkins at the time. And he was a postdoc. And he was working next to my hero. What's this guy's name? It's the only name you have to know in this course.
VBM.
Vernon Mountcastle. Vernon Mountcastle. Vernon like the soft drink, Mountcastle. That's the only name you have to know in this course. He's my hero. His name is Benjamin, not Byron. For 10 years I was teaching it as Byron, and then some sassy student who was on the internet while I was giving the lecture said, "Oh, Professor Barr, it's not that, it's this." His name is, the B stands for Benjamin, so it's Vernon Benjamin Mountcastle. He was doing studies in the somatosensory cortex years before these guys did any of these kinds of studies. And he was convinced, he was one of the first people to start using microelectrodes that could actually look at individual cells. And he was convinced that because there were 6 submodalities of somatosensation, don't worry about what that means right now, that maybe if you mapped it out through the 6 layers of the cortex and no one had any idea what their function was, that maybe they're segregated in terms of the laminar axis. In other words, if he found rapidly adapting mechanoreceptors or whatever in the first layer, he'd find something else in the second, etc. And he didn't. And he was very disbelieving that this could not be the case because he knew the anatomical organization was laminar, and he couldn't believe that these weren't segregated, that as he stabbed his electrode down, he'd find a given area of the somatosensory cortex that responded to a given area of the skin. And then what would happen is he would find that every single layer below that responded to the same area of the skin in the same submodality. In other words, they had the same functional organization all the way from the surface of the cortex down to the depth. And he refused to publish it because it didn't make sense. And he went up and down the halls of Johns Hopkins, scratching his head after lunch and such, saying, "God, it can't be true. This sounds like a columnar organization of the cortex, and it just doesn't make sense to the anatomy. I'm not going to publish this until I'm really sure it's true." And while he was walking up and down the halls of Johns Hopkins, refusing to publish his discovery that there was a columnar organization to the cortex. Weasel, who apparently knew him, Weisel, stole the idea. He got this idea, oh, it was a columnar organization of the cortex, maybe that applies to the visual system. And he skinnied up to Harvard and started working with Hubel, and they got the credit for discovering the columnar organization. This poor man, Vernon Benjamin Mountcastle, did not get the Nobel Prize. He got every other prize in science. He discovered, and even in the textbook now, they admit finally that he discovered the columnar organization of the cortex, and these guys essentially stole it. Poor man's dead now. He deceased in 2015, therefore he'll never get the Nobel Prize because they don't get to give it to you after you're dead, although they should. I think they should give it to you before you die because you can see what happens with a lot of the Nobel laureates once they get the prize. But he was the discovery of that. So they were right. I mean, they discovered this happened. Then through serendipity, they discovered, because their electrodes were sort of soft and wiggly, they discovered that if they could stick an electrode down and isolate a column, every once in a while the electrode would sort of skinny sideways and skinny more and more sideways. And then they realized, my goodness, if it goes sideways, these columns are juxtaposed to each other. And that if one column likes a line of disorientation, the one right next to it isn't random, it's a little bit more, then a little bit more, a little bit more. In other words, they're interdigitated columns, orientation-specific columns that make an orderly representation of all different little lines of different orientations. Yeah?
Am I just remembering this incorrectly, or are they also sensitive to the direction of the lines moving?
No, these aren't. That comes in a different area. There's sort of a progression of processing, but to the best of my knowledge, the simple cells that they found in this area of primary visual cortex was not sensitive to the direction the lines were moving. It wasn't motion sensitive. These were stationary signals that they were looking at. We'll get a little bit to that in a bit, okay? And then they also moved in the opposite direction. So they came up with this sort of idea of a hypercolumn. So there's on a hypercolumn, you have first little subcolumns of different lines of different orientation. And then if you move your electrode approximately in the opposite direction, you'll find out there's a whole nother slab next to it that does the same thing. The orientation columns are in register with each other, and that slab is from the other eye. So they came up with the notion that there are ocular dominance columns, slabs, and then subdivided in each one are little lines of different orientation. And then on the other axis, if you're trying to lay out this, they call this a hypercolumn. The idea of a hypercolumn is it was the smallest area within their conception at the time. It was the smallest area of tissue that could process all information they thought was important from a given area of the retina. Different lines of different orientation from both eyes or retinoids. And then if you want to go to a different area, you have to move over a little bit. So they envisioned the visual cortex as being a mosaic, if you will, retinotopic organization of hypercolumns, each one that can do all the processing that was required until you had to go to a different area of the retina. And the last dimension of the hypercolumn, which is true of all areas of the cortex, is the laminar dimension, where you have the input layer, the local processing layer, and then the output layer. So that was their sort of Cartesian view of what the smallest chunk could be. They could do all the processing within the cortex. And everything I want to teach you up to this point is true. It's just partially true. And then we're going to try to blow it apart a little bit later. You may ask yourself, well, how can you add that? They call these simple cells, by the way. They're in primary auditory cortex, area 17, the one that's most grouped around in Cocker and Fisher. And they ask the question, how do you make something that's sensitive to lines of different orientation when you know that the inputs to that area are all dots, which they've been looking for in the first place? Why couldn't we get any responses out of dots, but we can get responses to lines? And they came up with a simple idea, which is true. It has to do with convergence. So they would find out that this particular orientation doesn't light up the cell. This one, the opposite direction, really lights up the cell. This is it firing when it's being exposed to that orientation. And that dot stimuli have no effect really at all. And that the shape of the cells, which they call simple cells, the shape is sort of angular. It looks a little bit like a football, but it's elongated so that there's a center on and a surround off, or vice versa. They're about 50% distribution. And those simple cells are the first thing that they finally discovered, and that's how you get a linear responsiveness. And the way it's done is by convergence. So if you want a line of a particular orientation, say this simple cell, the way you do it is you take the inputs coming from the Nrsc, which are angular in shape, center surround, and you line them up strategically through interneurons onto a simple cell. And the simple cell, when it gets a line that falls across all three receptive fields at the right orientation, the cell really fires, and that's how you get orientation specificity. That there's convergence, and that's true. That is how they work. So when I start to tear that apart, don't think that that means they were correct. There is convergence from a much simpler representation of the visual field, little dots that go on in the retina and the LGN. A convergence on the individual simple cells that makes them sensitive selectively to now strategically organized receptive fields at different orientation along the retina, and then you respond to a different area of the retina. Absolutely true. There's nothing wrong with that theory. The problem is that they went on from simple cells, and they started moving their electrodes around, mainly to surrounding areas of visual cortex, area 17, which is adjacent to that. And they started finding cells that weren't that simple anymore. They seemed to like larger areas of the retina. They seemed to respond to lines, not just of certain orientation, but ones that had been moved into the field in a given direction. A certain length of line. They were sensitive to certain lengths of lines and not just always the same little line. And they thought, well, it must be that these cells are more complex than these simple cells are. And if you keep following that, and there's some truth to it, that progressive convergence is taking place again. The way you create these, what they call complex cells, is that you converge the outputs of a number of simple cells. And then they went further out, and there's some truth to that. And then if they went further out to area 19, I think it was, or maybe parts of area 18, and they found cells that were even more complex. They seemed to have even more response constrictions that would make them fire. And they figured that must be the convergence of complex cells onto other cells, so they called them hypercomplex cells. That's why they usually should probably kill people once they get the Nobel Prize, so they don't keep on going down these directions because they can't stop. Because they kept going. They came up with complex cells, hypercomplex cells, hypercomplex cells. What's wrong with that logic before we quit? If you keep on going through even the visual cortex, and you keep on saying that the visual world is firstly pixelated by the retina, and then as you start extracting higher and higher level, quote, higher and higher level features of the visual world, you have to have progressive convergence onto higher and higher level cells, where you're going to run into trouble. How far can this go? Doesn't it make you feel uncomfortable? You know, what's the next cell in line? You know, how much higher up can you extract? Can you get a cell that they can just recognize your grandmother? It would have to be eventually. Logically, if you think about the progressive convergence onto higher and higher level cells, you should be able to find a cell that responds to just your grandmother. And that's actually a joke in neuroscience. It's called the grandmother cell hypothesis. That this notion of progressive hierarchy without ever finding out that there must be a higher level code leads to a completely illogical conclusion, which is that this hierarchical organization lead to grandmother cells that just respond to very specific features. Then your grandmother with a red dress on. I mean, your whole brain would have to be full of grandmother cells in order to process what you're seeing right now. There's something wrong with it, and I want to leave that with you, and then we'll get to what's wrong with it and try to replace it in the next lecture.